Okay, so here we are. Here, just a second. Let me enable transcription because someone always asks for that. There we go. Live transcription enabled. And okay, someone was telling me there was a question about the exam. It's the Friday after we get back from break. It's still a little bit too early to give you one this coming Friday, and I'm not so evil that I would give you one the Monday after coming back from spring break. I mean, after almost 25 years teaching here, I'm pretty evil, but I'm not that evil yet. So let's, one more time, let's check the chat. Will there be a study guide for the exam? Thanks for the reminder. You've got the practice genetics problems, and I'll post a practice exam this afternoon. If I can dig up a more detailed study guide, I'll do that, but at least I'll give you some practice questions. All right, so where were we? We were talking about protein shape, and we talked about the 4 different structural levels that protein shape can be described on. The first is that proteins are polymers of amino acids, and there's 20 amino acids that are used to make proteins, and the amino acids have to be assembled in a particular order. That order is primary structure, and then portions of the chain will kink up into alpha helices, these spiral bits right here, or they will, there, I'm trying to use the highlighter right there. There's a nice little spiral bit, or they'll zigzag and form beta sheets like this thing that looks like an arrow that I've just circled over there. That's secondary structure. The entire thing will fold up into a complex shape. That's tertiary structure. And then for many proteins, they're made not just of one chain of amino acids, but several, each of which folds up, and then all of which have to fit together. And we call that quaternary structure. The reason this matters is that a living thing has to make a ridiculous number of types of protein. And proteins do a ridiculous number of things. They make your muscles contract and make it possible for you to move. They break down the food that you take in and convert it into forms that your cells can absorb, and then they do the work of building those nutrients back up. They run your whole metabolism. They do lots of different things. And for every protein to work, it's got to have a particular shape. And I've compared proteins to tools a lot. If you have a screwdriver or a socket wrench that doesn't have the right shape, then the tool can't do what it needs to do. The shape of the tool determines what you can use it for. Incidentally, just to try to bring this to something a little more concrete, if you apply something like heat or acid or sometimes alkali or other conditions, you can cause protein molecules to change their shape, usually irreversibly. We call this denaturing a protein. And a very simple example would be frying an egg. Egg white is made of proteins called albumins. And when they're raw, of course, they are translucent and kind of slimy. When you apply heat, when you fry an egg, you're not breaking the proteins down unless you burn the egg. But the heat causes the proteins to change their shape, and that causes the egg white to change from being runny and slimy and translucent to firm and opaque and white. So you denature proteins every time you cook an egg. If you were to take some milk and heat it almost to boiling, and then hit it with some lemon juice, the milk would curdle and you could strain out some curds. And that's actually one type of cheese. That's another example of denaturing a protein. Oh, another example, when you apply heat and certain chemicals to the proteins that make up your hair, you change the shape of the protein molecules, and that changes the shape of the entire hair shaft. And that's what you're doing if you ever get a perm. Or comb. If you ever get a permanent wave, you have denatured your keratin proteins that make up your hair. Denaturing the protein affects the protein's function, and usually it makes it no longer functional. So the function of a protein depends on its shape. The shape of a protein depends on the order that the amino acids go in. That's why all of this matters. You would not live if you couldn't make over 20,000 different proteins, each of which has one specific order of amino acids. So your survival depends on your cell's ability to put lots of amino acids together in exactly the right order over and over and over again. Before we go any further on that line, I want to talk about today one extremely important class of proteins. They're not the only ones that matter, but they're extremely important, and they're called enzymes. And to break down what enzymes do, I've got to talk a little bit about the energy of chemical reactions. We don't need to go into why atoms stick together. I've shown you diagrams in which atoms are just stuck together like ping pong balls stuck together with glue. Atoms do not really look like ping pong balls, and they're not really stuck together with glue. If you need to know what a chemical bond really is, we've got a perfectly good chemistry department that can handle that. For our purposes, we just need to know that when two atoms stick together to make a bond, it requires energy to make a chemical bond, and that chemical bond stores that energy. And when you break a chemical bond, you release that energy. When you set something on fire, what you're doing is causing all of the bonds that held those atoms together in molecules to start breaking, and that releases energy in the form of light and heat. And that's energy release. So if you burn a piece of paper, all of the bonds between the carbon, hydrogen, oxygen atoms that made up that piece of paper are broken, and energy is given off. However, you also need energy to form chemical bonds in the first place. It took energy to stick those carbons, hydrogens, and oxygens together to make paper. Ultimately, the energy source was the sun, which landed on tree leaves, and trees were able to harness solar energy to stick together carbons, hydrogens, oxygens, etc., to make the cellulose that paper is made of. And then when you burn a piece of paper, you're releasing that energy back into the world. So any kind of chemical reaction involves breaking and releasing and reforming chemical bonds. Bonds might break, bonds also reform. If a chemical reaction produces more energy than it consumes, the extra energy is given off, often in the form of heat and or light, and the reaction is self-propagating, and we call it exothermic. Simplest example there is fire. It only takes a spark to get a fire going, and once it's going, it will keep on burning until there's nothing left for it to burn. You don't have to keep lighting a fire once you've got it started. That's an exothermic reaction, giving off more energy than it requires. On the other hand, if a chemical reaction consumes more energy than it gives off, then you have to have a source of energy to make it keep going. And we say that that reaction is endothermic. Endothermic reactions consume more energy than they produce, and they need a source of energy to make them go. Now, here's the thing. We can draw graphs of the amount of energy that's available in a chemical reaction over time. And think about a fire. You have a bunch of wood in a fire pit with all safety precautions taken. Okay, we're not burning down a house or anything. We're lighting a campfire. There's lots of energy that's stored in the chemical bonds in the molecules that make up that wood. So we have a lot of what we call potential energy. There's a lot of energy that is stored that we can tap into. To tap into it, what we have to do is add a little bit of energy to get that fire started. We have to strike a match or, strike a lighter or something like that and get that wood burning. That little kick of energy that we have to give the fire in order to get it started is called activation energy. So to start a fire, we have lots of potential energy in those chemical bonds that make up the wood. So we have lots of it that is potentially available. To get it to start going, we have to give it this little kick of activation energy, this spark or this match. And once we get that fire going, it'll burn, and we don't have to add any more. You don't have to keep lighting a fire, and it will keep going until ultimately there's nothing left. And all of that potential energy in the wood has been released in the form of light and heat and motion, like the motion of, soot particles or sparks, things like that. And when the fire is finally done, all of that potential energy that was available in the wood has been released, and the ashes that are left have much less energy stored in them. So we go from a state where we have lots of potential energy up here, and then over time we have to add energy to it. We can think of that as going up a peak. And then when the fire really starts burning, it gives off energy. And we can think of that as going down a peak until finally we just have ashes that have less energy stored in them than the wood that we started with. And so the diagram that I've drawn looks a lot like a hill. Let's say we've got a ball at the top of a hill. That ball can spontaneously roll down the hill until it gets to the bottom. But let's say there's a little hump in front of the ball. To get the ball rolling down the hill, we have to give it enough of a kick that it can get over this hump. And then once it's at the top of the hump, it'll spontaneously roll downhill until it's finally at the bottom and doesn't roll any farther and comes to a stop. That hump that we have to get the ball over is called the activation energy. And where this comes into play in biology is that a substance that lowers activation energy is called a catalyst, and an enzyme is a biological catalyst. So a catalyst is anything that lowers that activation energy. A catalyst would lower or even get rid of that hump that we have to get the ball over. And a catalyst is not affected by the chemical reaction. If you're driving a car that is younger than the mid-70s, I can't remember the exact date, it might be 73. If you're driving a car that's younger than 1973 or so, and I assume all of you are, your car's got an attachment to the exhaust manifold called the catalytic converter. Your car exhaust passes through that. The catalytic converter has little strips of platinum in it, And as the hot exhaust passes over the platinum, compounds that are potentially dangerous, like carbon monoxide, are turned into less dangerous compounds like carbon dioxide. The catalytic converter makes your engine exhaust a bit safer and gets rid of some of the pollution that it would otherwise produce. And that's because the platinum in there acts as a catalyst. The platinum isn't used up. You don't have to go in for a platinum refill every 50,000 miles or anything.
That's why those get frequently stolen.
My wife says catalytic converters are sometimes stolen for their platinum because platinum is valuable. But there's not much in there, and you never need to refill your platinum because the platinum is not burned up by the chemical reaction that it does. The platinum is able to do what it does just by staying there. Enzymes act as catalysts in living systems. You don't need platinum in your cells, but you do need enzymes. And enzymes are proteins. And to work right, enzymes have to have the right shape. And a little aside here, there's a number of small organic molecules that aren't proteins, but play various roles in helping to make chemical reactions happen in your cells. And we call those coenzymes, and a number of vitamins, notably several of your B complex vitamins, are actually coenzymes. They're not actual enzymes, but they assist various chemical reactions to happen. So if a chemical reaction doesn't have an enzyme, then you may have to wait a long time for enough activation energy to come along to make it start. Right? If you've got a ball at the top of the hill, but the ball has to get over this hump, maybe eventually someday there'll be a wind that's strong enough to push the ball over the hump, but it might take a very long time. On the other hand, if we have an enzyme present, we get rid of that hump, or at least we lower the hump. And that makes it much more likely that that chemical reaction can start. Just like if we have this ball at the top of a hill and we just get rid of that hump, then the ball is freer to start rolling spontaneously downhill until it gets to the bottom. So enzymes make chemical reactions happen, and they do it by reducing the activation energy that's needed to get them started. Okay, how does this tie in with what we've talked about with shape? Molecules that an enzyme acts on are called the substrate of that enzyme. And an enzyme molecule may have a pretty weird shape, but there'll be a region called the active site, and the substrate will physically fit into the active site like a key fitting into a lock. Or if you like the head of a bolt fitting into a socket wrench. Once that substrate molecule is fitted into the active site, bound to the active site, the enzyme molecule might put stress on the substrate molecule and cause it to break, or the enzyme molecule might bring the substrate next to another molecule that also fits and stick them together. Enzymes can create chemical bonds as well as break them. And so when the substrate fits into the active site, the substrate might either break up or it might be joined with something else, depending on what the substrate is and what the enzyme is. And there's a diagram of how this works. The substrate is shown here as this kind of bright green, funny little shape. And the substrate comes down and fits into the active site of an enzyme. The enzyme might actually change its shape just a little bit to bind to the substrate. We call that induced fit. And then when the substrate is in the active site of the enzyme will put a little bit of strain and cause the substrate, in this case, to break. Once it breaks, the broken pieces of the substrate over here go off on their own merry ways, and the enzyme is still intact, and the enzyme can come around and do it again until all of the substrate molecules have been broken up into these products. So in this diagram, enzymes can break down. Enzymes can also build up. It all depends. And again, you could compare an enzyme to something like a socket wrench. The socket would be the active site. The rest of the wrench might not be all that important. You know, we could make the handle a bit longer or a bit shorter or a bit thicker, or we could paint it green and it wouldn't make that much difference. But the active site has to be a particular shape. Just like the socket has the shape that it does, and that means that it can only fit one size and shape of nut or bolt. The nut or the bolt here would be the substrate, and the socket on the wrench would be the active site of an enzyme. This is a model of an enzyme in your saliva. You actually start digesting your food the minute you put it into your mouth, because your saliva is not just water. Your saliva contains enzymes of a type called alpha amylase. And that's what's shown here in blue. This lumpy thing right there is yet another way of making a model of what a protein molecule looks like. And alpha amylase works on starches. When you put starch into your mouth, the starches, which are shown here in yellow, fit into this active site right here. So again, like a key in a lock or a bolt in a socket wrench. And they're broken down into simpler sugars. And you can actually feel this if you take something really starchy, like a cracker, and put it in your mouth and just hold it. At first, you're probably not going to taste very much. But as the alpha amylase in your saliva starts getting into that cracker, the cracker's going to get wet. And the amylase molecules in your saliva are going to start breaking the starches down into simpler sugars, and the taste of the cracker will change. You'll actually taste it becoming just a bit more sweet as you just hold it there. Try it sometime. So yeah, that is an enzyme that is as close to you as your own saliva. Just for fun, here's another real world example. This is another one of those models of proteins. The alpha helices are shown in green, like that one. And beta sheets are shown in red, like that one. And right here is the active site, and the substrate of this enzyme, beta lactamase, happens to be a molecule called penicillin. And when a penicillin molecule fits into the active site, beta lactamase puts a type of strain on the molecule and causes it to break. Penicillin-resistant bacteria are resistant because they're able to make beta lactamase. Bacteria that can make this enzyme can neutralize penicillin. They can break it down. Some of them can even use it for food. So this is why some people might get a bacterial infection, take penicillin, but not get better. This is something we've been seeing in increasing numbers. We talked about this a long time ago when I was talking about natural selection, that there are varieties of bacteria that are antibiotic resistant, and they've become more and more common over the years. And the reason those bacteria are antibiotic resistant is they're able to make beta lactamase, which breaks penicillin down and even breaks it apart in such a way that the bacterium can eat it. Okay, a lot of the metabolism that your cells have to do is long series of chemical reactions, not just a single chemical reaction, but many steps, a lot like an assembly line. If you think of an assembly line to make some sort of gizmo, you may have many machines, and at every step, only one step takes place. If you've got a car on an assembly line, at one station, a robot will go zip, 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 and install maybe 3 bolts. And then the chassis goes on to the next step, and a different robot will go zap, 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 and install 3 different bolts. And then we go on to the next step, and a third robot will go whiz, whiz, and install 1 widget. I don't know. I have never actually worked in a car factory, but a lot of your metabolism is done on the principle of the assembly line. And I'll give you a small example of this that I hope does not hit too close to home. Let's say you take in some ethyl alcohol. This is the alcohol in alcoholic drinks. It's occasionally present in certain foods as well, such as very ripe fruit. Well, ethanol has various effects on your body and your nervous system, including a tendency to dance on tables with a lampshade on your head. And your body does need to break it down because alcohol is ultimately toxic. Too much of it in your body, and you'll end up with a slight case of the death. So your liver cells make an enzyme called alcohol dehydrogenase. And alcohol dehydrogenase will bond to ethyl alcohol and turn it into a compound called acetaldehyde. Now, acetaldehyde is still not really good for you. In particular, it causes various symptoms such as headaches and skin flushing and feeling like you've just been run over by a truck. Acetaldehyde is actually responsible for a number of the symptoms of hangovers. Your body still has to get rid of it. So a second enzyme called aldehyde oxidase breaks the acetaldehyde down and turns it into acetic acid. Now, acetic acid is not particularly toxic. It's the active ingredient in vinegar. So if you've ever had a nice vinaigrette salad dressing or something like that, you have eaten acetic acid. And acetic acid your body can deal with. Acetic acid can just be excreted. So we have this 2-step process to convert alcohol into a product that your cells can just flush away. And if we diagram it like this, that's what ethanol looks like. That's the ingredient in alcoholic beverages. And an enzyme, alcohol dehydrogenase, first converts ethanol to acetaldehyde, which looks like that. And acetaldehyde is still not really good for you. So a second enzyme called aldehyde oxidase converts acetaldehyde into acetic acid, which again is the active ingredient in vinegar, and which is not particularly bad for you and which your body can just dispose of. So there's a 2-step process for detoxifying alcohol, and your liver cells do this all the time. Hopefully they don't do it too often. Please drink responsibly and in moderation. Here's where things get fun. This is an edible mushroom right here. It's called Coprinus atramentarius, otherwise known as the inky cap. I have seen it growing around here, and it's edible. Tastes pretty good if properly prepared. But the problem is that it blocks aldehyde oxidase. If we go back here, if you drink alcohol and you eat those mushrooms, this step right here is blocked, and your cells can no longer turn acetaldehyde into acetic acid. So if you eat those mushrooms and then you drink alcohol, the alcohol gets converted to acetaldehyde, but there's nowhere for the acetaldehyde to go because you can no longer turn it to acetic acid. It's like a blockage on the assembly line. And so the result of eating these mushrooms and drinking alcohol is an instant hangover from hell. You build up acetaldehyde and you can't get rid of it. And so you feel absolutely wretched. So these are perfectly edible and even nutritious mushrooms, but you cannot serve wine with the meal. You can't drink alcohol for about 24 to 36 hours after you have eaten these mushrooms, or you will feel like you've been hit by a steamroller. And incidentally, there's some prescription drugs that do the same thing. Antabuse is one. There's another one, calcium carbamide, which is sold under the name Tempacil. And they're used to treat alcoholism because if you take these, they make the side effects of drinking extremely unpleasant. You get a wretched hangover before you even start feeling drunk, as long as you've got these drugs in your system. So they can't really stop you from drinking alcohol, but they make it utterly miserable, which makes it a lot easier to quit. And they work by blocking those chains of chemical reactions that are driven by series of enzymes. So there's an assembly line. And as I've said, you could think of metabolism as being like an enormous set of assembly lines building up and breaking down molecules. And every enzyme would be like one of these yellow industrial robots that performs just one step and one step only. Everything okay, sweetie? Okay. All right. And if we diagram out the basic metabolism that all living cells do, it looks like this. I'm not expecting you to memorize this, of course. It is supposed to look like a rather messy plate of spaghetti, but every one of those little black lines that you see is one chemical reaction. And every one of those black lines is catalyzed by an enzyme. And all of these reactions are what your cells have to do in order to process the nutrients that they get and convert them into the molecules that it needs. This is everything your cells have to do to carry out metabolism. It's a lot. And you have to make many thousands of different enzymes in order to support your life, in order to maintain that metabolism for as long as you live. This is the point where I'm imagining you all going, "Oh, dude." I'll show you a small part of these reactions, and I promise we'll end with something fun. What's diagrammed here is a set of chemical reactions called glycolysis. Your diet includes some sugars, and even if you're not eating sweet food, the starches that you eat in your food get broken down into simple sugars, the most important of which is a sugar called glucose. Glucose does taste sweet, but not as sweet as table sugar. You may know that glucose levels have to be maintained at within certain limits within your bloodstream. If you have diabetes, you may have excessive glucose in your blood, and you have to monitor this. And a small but critical part of metabolism involves breaking glucose down into smaller molecules. So every substrate here is printed in black, and glucose right here is up at the top. And glucose has to be converted to glucose 6-phosphate by an enzyme called, I can't actually read that because I've got a little notice up in front of it. And then the glucose 6-phosphate has to be converted into fructose 6-phosphate by an enzyme called phosphoglucose isomerase. And then the fructose 6-phosphate has to be converted into fructose 1,6-bisphosphate by an enzyme called phosphofructokinase. And then that fructose 1,6 bisphosphate gets split in two by an enzyme called aldolase. And the two fragments are converted into glycerophosphate by an enzyme called triose phosphate isomerase. And then that's converted by GLAP hydrogenase into 1,3-bisphosphoglycerate. Ultimately, you end up with two molecules of a compound called pyruvate at the very end, after 4, 5, 6, 7, 8, 9 different steps, each of which is catalyzed by a different enzyme. Ultimately, you don't need to pay attention to what's in blue, but ultimately, this is the way you get chemical energy stored in your cells, because you make molecules of an energy storing compound called ATP, which I will talk about later. Let's not worry about it right now. Right now, just know that you have about 9 steps to get from glucose in your food to a couple of molecules of something called pyruvate. This is a yeast. This is a single-celled fungus. That's the definition of a yeast, seen under a microscope, and it happens to be called Saccharomyces cerevisiae. And what this yeast does is it takes pyruvate and it carries out a set of reactions called fermentation. And when you ferment pyruvate, the yeast converts it and gets a little energy for its metabolism out of it. The yeast converts pyruvate into ethyl alcohol, the stuff in alcoholic beverages, and carbon dioxide. And fermentation is another one of these multi-step processes that requires an enzyme for every step. And so if you take some Saccharomyces cerevisiae yeast and you turn them loose on a sugar-rich liquid, such as grape juice, let the yeast do its thing, and boom, shalaka-laka, boom, you've got wine. If you do it with other fruit juice, you've got fruit wine. If you do it with diluted honey, you've got mead. You can make various alcoholic drinks this way by taking advantage of yeast fermentation. Incidentally, it also works in bread. If you add yeast to bread, yeast will break down the sugar in your bread dough, I should say. If you add yeast to dough, yeast will break down the sugar, they'll convert it into alcohol and convert it into bubbles of carbon dioxide. The bubbles that the yeast make your bread light and fluffy. The alcohol is driven off in the baking process, so you don't have alcohol in baked bread. In theory, you could get drunk by eating a lot of raw bread dough, although in practice, you probably just make yourself really sick. Don't actually try it. Okay, one more thing. This is barley. It's a grain called Hordeum vulgare. Not a lot of sugar is present in barley, but barley does have a lot of starch. And barley grains are seeds. And when a seed is planted, it needs to have a source of energy so that it can grow until it can get its leaves out above the soil and start doing photosynthesis on its own. So the nutrients that are inside typical seeds, which would include grains and also nuts, are nutrients intended for the use of an embryonic plant. And in the case of barley, that means there's a lot of starch in barley seeds. Here's the thing, you can soak barley in water and then spread it out and let it dry. And that's what this guy is doing. All that golden stuff is barley seeds that have been soaked and spread, and they're drying. And if you do that, every seed begins sprouting. And right as sprouting begins, every seed begins to make enzymes. Enzymes that will break down the starch and turn it into sugar. And by the way, barley that has just begun to sprout and has just beginning to break down its starch and turn it into sugar is called malt. So this gentleman right here is making malt. He is a maltster. That's his title. Okay, remember that starch is a polymer of simple sugars. And diagrammed in blue here, we have molecules of an enzyme called amylase, similar, although not quite the same, to the amylase in your saliva. Amylase enzymes break down that starch, which is shown in light green, into simpler units called maltose, which is a sugar which the seed would normally use as an energy source for its earliest growth. Normally, the embryonic plant inside the seed would take that maltose and break it down and use it as an energy source for itself. However, that malted barley, once you've got it going, we can take that malted barley, grind it up, and soak it in hot but not boiling water at about 143 Fahrenheit. That's what, 70-ish Celsius. I can't be bothered to do the conversion. And if you do that, it creates a sweet liquid called wort. And then we take the wort and we feed it to our yeasty friends, Saccharomyces cerevisiae, let them do their thing on the sugars in the wort, and the end result is beer. Beer retains traditionally the alcohol from the yeast fermentation and also the carbon dioxide gas as well. Mass-produced beer, they filter out the yeast and then pump carbon dioxide into it to make it fizzy. But if you've ever had homebrewed beer or what they call cask-conditioned ale, which you might have at some of the fancier shops, fancier stores, that beer will retain the carbon dioxide that the yeast naturally produce. And I have just told you the basics of how to make beer. There's a store in North Little Rock called Fermentables. If you want to try it yourself, they can sell you the things you need. But whether you do it or not, whether you even drink beer or not, regardless, what you need to know is that beer is made possible by humans' ability to harness enzymes that living cells produce and make use of. Both enzymes in the barley that turn the starch to sugar and enzymes in the yeast that turn the sugar into alcohol and carbon dioxide. Not that you asked, but you can make stronger drinks by brewing basically a beer, albeit not one that you'd really want to taste, or a corn beer, or a wine, and then distilling it to concentrate the alcohol. And then often we store the distilled alcohol. You know what distilling is, right? That's where we heat the stuff and we collect the alcohol as it evaporates. And when it comes straight out of the still, it is, of course, called moonshine or white lightning. But if you store it in a wood barrel for a long time, you'll mellow out the flavor and you'll end up with whiskey or bourbon or a nice brandy or something like that. So there, see, this information might seem like a bunch of trivia, but it's actually incredibly useful, especially if you're going to be a moonshiner. Never let it be said I don't give good career advice. On that note, I will now stop the recording.